Assalamu alaikum everyone, myself Sana Nawaz. Today I will talk about the major scientific discovery that won 1989 Nobel Prize in Physiology and Medicine. It is about the discovery of retroviral oncogene made by two brilliant scientists, Howard E. Wormus and J. Mitchell Bishops. This discovery changed the way we understand cancer and opened a new path for research and treatment. Let's meet the minds behind this discovery. First, we will discuss about Howard E. Wormus. Howard E. Wormus was born in 1939. He is a famous virologist. He earned his medical degree from the Columbia University and pursued his research career at National Institute of Health. He is famous for his study on the genetic mechanism of retroviruses. On the other hand, Mitchell Bishops was born in 1936, obtained his medical degree from the Howard University, and became a leader in studying how genes can trigger the cancer. RSV stand for reverse sarcoma virus is a type of retrovirus that was known to cause cancer in chickens. A retrovirus is a virus that inserts its genetic material into the DNA of the host cell. Scientists believe studying RSV virus might reveal how viruses can cause the cancer, which could provide clue about cancer in humans. उसके बाद उन्होंने क्या किया, उन्होंने इस वायरस को आइसोलेट किया. उन्होंने इस वायरस को आइसोलेट कैसे किया? वर्मस एंड बिशन टुक अ ट्यूमर सैंपल फ्रॉम अ चिकन इन्फेक्टेड विद RSV viruses. They isolate the virus from the tumor and focus on its genetic material. By analyzing the virus, they wanted to understand how it could change normal cell into cancerous cell. In lab, they grow cells infected with RSV to observe changes. They notice that the infected cells start to divide uncontrolled as a key sign of cancer. The research found that RSV genetic material carried a special gene which they name as SRC, pronounced as sarcoma. The gene was different from those in other retroviruses. The SARC gene, SRC genes, was responsible for making cell grow uncontrolled, leading to cancer. In this slide, we will discuss about the mechanism of action, that how a cellular gene switch for a cancerous gene. Wormus and Bishop discovered that the SRC gene, which caused cancer, was not originally a part of the viruses. Instead, a similar version of SRC gene already existed in normal cells. The gene was part of chicken's DNA and served as a normal function in the cell. They name as cellular gene CSRC gene, where C is stand for the cellular. The SRC gene is a proto-oncogenes, meaning it's a normal gene that help regulate cell growth and the division. Proto-oncogenes are like on and off switches that control when the cell is grow, when the cell divide, or when the cell is stop growing. The viruses RSV had stolen the CRSE gene from the chicken cells. Over a time, the virus altered this gene, turning into a mutated version called VSRC gene, where V stands for the viral gene. The effect of the mutation caused the SRV gene act like a stuck gas pedal in the car. It made the cell grow uncontrolled because it is no longer responds to normal growth signal. This uncontrolled growth caused the cancer. In this slide, we will learn the significance of this discovery, understanding the cancer roots, the cellular basis of the cancer. It is proven that cancer can start from changes in normal genes, not just from like smoking and infections or other external factors. New treatment approaches, their work paved at the way of developing new cancer treatments. Understanding the role of oncogenes open us possibility for targeting specific genes involved in tumor development. Foundation for future research. To inspire scientists to find other genes linked to cancer in humans, the discovery of retroviral oncogene laid the groundwork for future research in cancer genetic and molecular biology. It led to the identification of many other oncogenes involved in various human cancers. What are the future scope of this discovery? Their work set the stages for the future. Number one, targeted treatment, creating medicine that attack cancer causing genes without harming normal cells. Number two, understanding cancer growth, learning how cancer develop over time. Number three, discovering new genes, found more genes that might cause cancer. Number four, genetic editing, using tool like CRISPR to fix genetic mistakes that lead to cancer. Moving forward, to understand how their discovery changed the world. Unrevealing cancer genetics. Their discovery opened the door to understanding the genetic basis of cancer and revolutionized the way we think about cancer development and treatment. Advance in cancer therapies. Their work led to the development of target therapies that specifically target oncogenes, offering new hope of patients and various types of cancers. They inspired a new scientist generation to keep studying on cancer to finding the solutions. To wrap up, this discovery by Holland and Bishop is a perfect example of power of curiosity and determination. They didn't just uncover the cancer work, they gave us hope for better treatments and a bright future. Their legacy continues to inspire scientists and change the lives. Thank you for your listening. If you have any question, I would be happy to answer them.